please welcome to the stage Dr. Gary Lyman, who will be speaking on biosimilars and oncology clinical pathways, perfect together. This session is supported by an educational grant from Coherus Biosciences and Pfizer, and there will be polling questions for this one.
Thank you so much. Obviously, we've dwindled in numbers, but that's to be expected. My job is to make sure you miss your plane this afternoon, and then hold somebody else responsible. But thanks for joining. So, as was mentioned, I will be discussing biosimilars and their role in terms of integration into clinical pathways. And I hope to convince you over the next few minutes that this is a good fit, and it makes a great deal of sense to integrate biosimilars. But I understand there remain concerns, apprehensions, and some level of uncertainty about what these are and how they're approved and whether they're really safe to use in clinical practice, whether they're in pathways and guidelines or not. So we'll go forward with this. I believe I'm Gary Lyman at the Fred Hutch and University of Washington. These are my disclosures, and in addition to some consulting, I chaired the American Society of Clinical Oncology ASCO working group on biosimilars, and I'll refer to that as we go through the presentation. These are the learning objectives. I was told not to spend any time on this because you've probably seen them. So in way of introduction, I don't need to convince you and hopefully your colleagues that biologic therapies derived from living systems have really revolutionized, I would say, medicine in general, many areas of medicine, but most notably in clinical oncology and in the treatment and care of cancer patients. With the beneficial effects of biologics, while they're clear from randomized controlled trials, and we've talked a fair amount about trials in this meeting, the economic impact of these medicines has placed an enormous financial burden on patients, families, and society. These are very expensive medicines. You've heard that time and again, and the burden on patients has really climbed precipitously over the last decade to decade and a half. Such what we're calling financial toxicity may limit drug access to these exciting and potentially curative biologic therapies and may result in some patients who could be cured, perhaps stopping treatment early with potentially devastating consequences. One strategy that's been proposed in legislation by the FDA, supported by the White House, the President's Cancer Panel, virtually a broad spectrum of support for the introduction, development, regulatory approval, and integration of biosimilars into clinical practice. And this, again, has come with some complexity, and that's what we're going to dive in today. But the primary goal here is to bring competition and bring down the price of these expensive drugs, and as an aftermath, improve access and reduce the financial burden on patients with cancer. So, and at the end, we'll talk about there are now 23 FDA-approved biosimilars in the U.S. There are actually many more in Europe, and I'll mention that briefly. These are based on far more preclinical analytic, looking at structural and functional characterization using a vast array of analytic tools. Some preclinical data in terms of animal safety and efficacy testing, and then in humans, more limited data than we're used to seeing with drug approval in the U.S., including the originator biologics. So there may be, there always will be PK/PD data, some immunogenicity testing, but not the requirement for multiple large randomized comparative trials that we've grown used to. And this is what I think makes some concerned whether efficacy and safety of these agents is comparable and non-inferior to the originator biologic therapies we've come to know and love over the last 10 to 20 years. So why the need for biosimilars? This should be quite intuitive, both from my comments and from your experience. That is, the cost of medical care in the U.S. has for decades now exceeded that of the increase in the gross domestic product. Nowhere is that more evident in the rise in cost of cancer care, cancer medical care, and even more so, the rise in the cost of cancer medications, cancer drugs, and these the poster child for this are the biologic therapies. So I show on the right-hand side the top Medicare spends on drugs, and if you look down this list, 7 of these 10 top agents are agents that we use in oncology. So again, this is a big driver of this increase in the cost of healthcare. Another way to look at it is to look at the expenditures for U.S. medications, and you can see that 30%, nearly a third of the expenditures on medical care in the U.S. is from these top 5 therapy areas, and number 1 on the list is oncology. One other way to look at this is the price of these drugs and what's happened to them, the price at or shortly after FDA approval. The data out of work from Memorial Sloan Kettering looking at the cost of cancer drugs at the date of FDA approval. And you may not see it there, but there's an orange line that shows the median rise, which now is exceeding $10,000 per month in terms of the cost of cancer drugs that are approved by the FDA. And you see the scatter plot. Some of these are far more than that median. So biologic therapies, both in the U.S. and globally, the spending on these has increased dramatically also over the last 10 years or so. You can see global biologic sales up through 2017. In 2018, data has continued on this trend, well over $200 billion globally. They continue to outpace overall spending growth in other areas, and now have basically reached about 20% of the global market value. This raises these concerns we'll come back to about whether this is going to limit, complicate patient access to these drugs, which again can be potentially curative or prolong survival, or leave them or their families in enormous financial debt. One final way to look at this is at the patient level, and this is an interesting graphic that I think says a lot. And the issue is that median household income in the U.S. has remained essentially flat. Some indications maybe it's even gone down in terms of effective purchasing power. At the same time, and that's shown in the red line, horizontal line. At the same time, the median monthly cost of new anti-cancer drugs in the U.S. has risen precipitously since the mid-'90s and has long surpassed that of the median monthly household income for U.S. households. So, again, there's no indication that this trend has slowed very much, and I think we can say without a doubt that this is posing an enormous challenge for a large proportion of patients with cancer under our care. So what are these biosimilars that we're hoping will help? Obviously, it's not the whole solution. We have lots of structural problems within our healthcare financing situation, but it is hoped that the biosimilars will be a piece in the puzzle of solving some of these dilemmas. So the FDA has formally defined a biosimilar as a biologic product, like the originator biologic, but one that is highly similar, parenthetically not identical, but highly similar to a U.S. license reference biologic product, for which there are no clinically meaningful differences in safety, purity, or potency. So this is a formal definition, and of course it requires some actual implementation. But I think the best way to think of this is comparing these biologic therapies in general, and biosimilars included, on the right-hand side, illustrated by a monoclonal antibody, to simple, small chemical agents that can be synthesized in the laboratory precisely, exactly. We have generics. Their structure is completely well defined. It can be reproduced, completely characterized. These are generally pretty stable molecules, and with few exceptions, they're not immunogenic. So the monoclonal antibodies or biologics in general, on the other hand, are very large, complex agents with that have a high molecular weight. They're produced in living systems, so again, they can't be replicated exactly due to variability in the biologic processes and the processes used to develop them. Many of them are relatively unstable and sensitive to external conditions. And again, because these are large proteins, they may be immunogenic. So stark contrast to agents going back a couple of decades that we and still use today. So we published a paper in the New England Journal about a year ago to basically try to summarize the evidence around these agents and educate both general practitioners and oncologists on the opportunities and reality of biosimilar medications. The requirements the FDA has established for biosimilarity are fairly simple and succinct. The biologic product must be highly similar to the reference product, notwithstanding minor differences in clinically inactive components. At the same time, there can be no clinically meaningful differences between the biologic product, the biosimilar, and the reference product, the originator, in terms of safety, purity, and potency. And the process for getting approval of a biosimilar that the FDA follows is one that we consider to be stepwise evidence development. And that's illustrated on the right. So again, the base is where a great deal of the focus goes, and we now have well over 60 analytic quality metrics that these agents must go through and demonstrate that they're highly similar to those reflected by the originator in terms of, again, structure and functional characterization. And then there's preclinical data. Again, this is done generally in animal systems. And then the clinical data is heavily done around pharmacokinetic, pharmacodynamic, and immunogenicity. And much of this is often done in healthy volunteers because these are generally individuals who are not immunosuppressed and are probably going to be more sensitive to reflecting some of the potential toxicities or the pharmacokinetic pharmacodynamic changes that may impact on the distribution of these agents. And then, if needed, and to some extent, there will be clinical data, often some comparative clinical data between the biosimilar and the originator. But the goal of that is merely to look and say, is there still uncertainty if the biosimilar is not truly highly similar to the originator? And then the FDA has the discretion to require or impose additional clinical trials. And again, we'll come back to this. A little easier to see in this stepwise process. We start with the analytic studies, again, looking at the structure of the biosimilar and its functional characterization. At each step as we go up, the FDA evaluates the totality of evidence that comes out of the previous step and determines whether further studies are needed. There's always a need for some animal studies in this process, and then early human studies looking at PK/PD and immunogenicity. And then, as I said, if residual uncertainty remains, there may be a requirement for additional clinical studies, including comparative randomized trials, but generally smaller trials, often phase 2 studies, not multiple large phase 3 trials. So there are a few new concepts, or at least not totally new, but ones that I think are unclear to many of my colleagues as they talk on this subject around the country. One of these is this concept of extrapolation. Generally, the approach here is a company develops a biosimilar, decides on what label, what indication they're going to go for, and does both the preclinical and clinical studies necessary to demonstrate high similarity or a biosimilarity to the originator. And if things go well and the FDA approves it for that indication, it can be marketed for that particular disease or clinical situation. However, many biologic therapies, as you know, have multiple indications, multiple diseases, or multiple scenarios of a disease, early stage versus advanced stage disease, and so forth. So the act that established the process for biosimilarity gave the FDA discretion to extrapolate based on the evidence presented for the initial indication and any additional scientific justification for granting extrapolation to other clinical scenarios. And if the FDA is satisfied, again, they have the discretion to grant extrapolation without, again, requiring additional clinical trials to establish the efficacy and safety of that agent in these other settings. Compare that to what the originator had to do for each new indication, each new label. A whole new set of data, clinical data, had to be submitted to grant that other indication. But for biosimilarity, the argument being that if you required all that data on each new highly similar agent, the cost of developing those agents will be at least as much, maybe even more than the originator, and you wouldn't accomplish the one goal, the major goal here, of developing these agents at a lower cost, allowing them to price reduce and compete with the originator. Now, there are a number of steps, again, a company needs to go through to establish the justification for extrapolation to the FDA. And it is important to note that extrapolation is not an automatic thing. And you will see already biosimilars on the marketplace that were not granted extrapolation for other situations. So again, the biosimilar has to be given approval for its initial submitted application and indication. In addition, however, they may be granted extrapolation if they can demonstrate that the mechanism of action for the other indications is essentially the same. Their binding, molecular signaling is essentially the same. The location, the receptors for these different indications the same. And then with PK/PD data, they can demonstrate that and support that the mechanism of action of the agent for these other indications is essentially the same. Now, in addition to that, however, there are possibilities that a different indication in a different population would reflect or express different toxicities, or patients may have different set of comorbidities or concomitant medications. So if the situation, if the patient population or the conditions of use are sufficiently different than the original indication for which they achieved approval, the FDA may say, we won't grant you that extrapolation until you provide us with more data. So again, they have the discretion, but it's not an automatic thing. The other major issue here, and we've already alluded to this, is biologic agents, including biosimilars and originator or reference products, are sources of variability. These drugs, these agents, because they're produced in living systems, they undergo fermentation conditions, different raw materials, protein purification methods may differ. There's all sorts of steps that kind of illustrated in this diagram on the right-hand side here, where variability can creep into the process. So even though you start off with the same vector, you start off with the same cell system, you try to do things exactly the same, there may well be differences in, again, structure or even functional characterization of the biosimilar in the end. So all this has to be monitored. It has to be demonstrated by the company that the agent is again highly similar within the boundaries that the FDA has established. And then over time, and we'll get into this more, there is the potential for drift occurring. Drift being that because of changes in manufacturing, the components that go into that process, the geography, there may be some changes, unavoidably going to be some changes in these molecules over time. And if those changes affect critical areas in the molecule, in this complex protein, there could be a change in efficacy or a change in safety. So there's a requirement not only up front to demonstrate high similarity to the reference product, but then over time, anytime you change manufacturing facilities or processes, you need to redemonstrate to the FDA that critical functionality, the high similarity of these agents has not changed in a clinically meaningful way. I would emphasize, however, and again, I'm going to illustrate this in a second, that both the original biologic therapy and the biosimilar are subject to this drift. In other words, they're both fundamentally complex proteins, large molecular weight proteins, with complicated manufacturing processes that you saw, and they're both subject to this drift and therefore both requiring continuous monitoring for efficacy and safety. So I'm going to illustrate this with a project that I've been involved with the last couple of years that's, I think, both concerning but hopefully eye-opening and a warning flag to us that we really need to monitor these agents over time. So one of the biosimilars that the FDA has approved undertook all the necessary processes, analytic characterization of structure and function, PK/PD studies, and then did comparative clinical trial between the originator, this is for the originator, Herceptin, and the biosimilar trastuzumab that they were trying to get FDA approval for. And in that trial, they found, to their surprise and somewhat shock, that the biosimilar results seemed to be superior to the originator. And they're puzzled. All the PK/PD studies showed high similarity. It seemed to be the same molecule to the originator, but when they ran the clinical trial, there seemed to be some superiority. And in fact, the FDA will not grant biosimilarity status to an agent that is demonstrated to be superior to the originator, something you might call bio-better. It has to be within these boundaries, and they're kind of illustrated here for this one performance characteristic. Again, there are 67 now currently in the FDA portfolio of quality metrics that these agents go through. But for each of these, they should fall within these boundaries to demonstrate comparability for that metric. So this one here is for ADCC, for Herceptin or trastuzumab. So it's antibody-dependent cell-mediated cytotoxicity. Some still believe that this is a critical action of trastuzumab in terms of its anti-cancer activity. But when they went back and said, we got to figure out why the biosimilar is doing better than the originator, they looked at all these characteristics, but what they did is they obtained multiple batches of the originator over time. And what you can see illustrated here is, if I can go back here, is that there was a drift. And this is the dots here on this graph represent European version of Herceptin, U.S. version of Herceptin, and then Herceptin used in the clinical trial. So there are different colors here, but they all show the same thing, that in recent batches, there was a drift where the ADCC performance fell below FDA criteria for that metric. So the question is, does that matter? How important is it? And does it explain why there seemed to be some advantage to the biosimilar over the originator? So they continued their trial, and we just published in European Journal of Cancer the 3-year data. We now have 4-year data on this, which has basically shown the same thing, that patients who received the biosimilar, which is shown, it's a little hard to see, but it's the top curve here. It's the blue curve, so it overlaps with this red curve, which represents the originator that had not drifted. So patients in the trial who received either the biosimilar or the originator Herceptin, but undrifted versions, did the best. But patients who received one or more batches of the drifted product seemed to have a greater rate of recurrence and a greater mortality rate. And these reach statistical significance. But I would caution you that this is retrospective subgroup analyses. We don't have data on all these patients. And this was not an a priori hypothesis that was raised. But it does, I think, bring to mind the fact that both the originator and the biosimilar can drift over time if we're not particularly cautious in monitoring these. And this study will go out for at least for 5 years, and we'll see if those differences remain. Now, again, somewhat reassuring is that in the most recent batches, there seems to be a recovery of ADCC performance in the originator. So somebody figured it out and got things on top of it. But again, this is a message that drift is, up until now, has been a hypothetical concern, largely for the biosimilar products. But it's, number one, a thing that can happen to both originator and biosimilars, and number two, it's no longer hypothetical. It can occur, and it has occurred in time. And if that drift involves critical functionality of the molecule, it may impact on patients going forward. So finally, this issue of immunogenicity, of course, it's a concern of all biologics. Again, these are large complex proteins. If it involves a neutralizing antibody or involves a critical portion of the molecule, of course, there can be loss of efficacy. And of course, there's always this concern or possibility of an allergic response, sometimes severe anaphylaxis. So immunogenicity is something that's tested at the very beginning and needs to be monitored throughout changes in the development and implementation of biosimilars. So the last thing, and this is a concept which I think is completely foreign until recently, and that's interchangeability and substitution. And the legislation that established the Biologic and Biosimilars Price Protection and Innovation Act said the FDA had the authority to establish a higher level of approval beyond biosimilarity, and that is interchangeable biosimilarity. And in turn, CMS could implement reimbursement or rules for compensation that reflected that. Well, to date, there have been no products that have been requested to obtain interchangeability status. So no companies come forward and requested interchangeability. So biosimilars have to first establish biosimilarity, but then they would need to provide additional clinical data, and that's just not been done. However, if a company comes forward with data and the FDA says, you're not only an approved biosimilar, but you're an interchangeable biosimilar, the rules state that they could grant substitution with that interchangeable biosimilar without the intervention of the provider. In other words, you could order the originator or a different biosimilar, and an interchangeable biosimilar could be substituted. Now, again, caution that, again, none have been so submitted or approved. And number two, that state substitution laws, and now well over 35 states have passed laws that would presumably preempt this, that would prevent this automatic substitution without notification. Can't prevent the automatic substitution necessarily if it's an interchangeable, but there'd be a requirement for the provider to be notified. So that's potential in the future, but I think it's important to understand. Now, the nomenclature here has confused a lot of folks, and I think just quickly review it here. The reason for nomenclature that's been established is the importance of tracking the specific agent, the specific biosimilar over time in terms of pharmacovigilance. Yes, we feel very confident in the processes put in place that the biosimilars that have been approved are safe and efficacious for the indications given. But rare or delayed effects may occur, and we want to make sure that we can trace any adverse event, unexpected, or loss of efficacy back to a specific agent. So the process that's been put in place, and there's a lot of consternation about this, is that the names of these agents have two components. There's the core name, the non-proprietary name that you see here in blue on the right-hand side. And then there's a random 4-digit suffix to that identifies the specific biosimilar that patient presumably receives. These are random with the exception of this middle one, SNDZ, which was approved before the nomenclature process was put into place. So you can infer which company makes that particular agent. And that was the first biosimilar approved in the U.S. But then these rules are put in place. For all future biosimilars, there'll be the somewhat arbitrary or random 4-letter suffix imposed. And you just have to become familiar with them if they're ones that you're using in your practice. So where are we currently with biosimilars in the U.S.? Well, the starting point is find out when originator biologic therapies patents expire. So we list here that biologic cancer treatments with more than $20 billion in global spending targets biosimilar development. Many of these have expired over the last 4 or 5 years. You can even go back to Politan Alpha, which expired before that. In Europe, you see at the bottom, but let's just look at the U.S. The pegfilgrastim patent expired in 2015. You see the anti-cancer biosimilars patent expirations have been more recent, and thus the release of these and approval and release of these is more recent as well. So here's a list, and I'll show you a little simpler list in a second, but of the approved biosimilars in the U.S., and you see how rapidly these have come along. So again, going back to the far left-hand side, you see that filgrastim SNDZ, supportive care, white cell growth factor, that was approved in 2015. The first true biosimilar. There was a agent that was approved as a biosimilar in Europe, but went through an originator pathway because the FDA hadn't established its regulatory authority over biosimilars, and that's tibofilgrastim. But that's technically not a biosimilar by FDA definition. And then, of course, there are other biosimilar supportive care agents, filgrastim, pegfilgrastim, that have come along. And then over the last couple of years, we've seen some of these large monoclonal anti-cancer therapy biosimilars come along, including bevacizumab, trastuzumab, and rituximab. So these are now multiple biosimilars that are out there. And I'll just show you kind of looking at the left-hand side are the supportive care biosimilars in the EU, in Europe on the left-hand side, and in the U.S. And you can quickly see how farther ahead Europe is in terms of regulating and approving these, going back nearly 10 years. The FDA approvals, we now have 2 filgrastim biosimilars. We have 2 pegfilgrastim biosimilars. We also have an potent alpha biosimilar. It's not on here. And then on the right-hand side are the anti-cancer biosimilars. And we have 2 rituximab biosimilars for lymphoma. We have actually 5 trastuzumab biosimilars. I mentioned one earlier with that issue around drift. But there are now 5 biosimilar trastuzumabs and an originator, so 6 choices there, and 2 bevacizumab biosimilars. Not all these are on the shelf at the moment. There are patent disputes that some of these have been embroiled in, what we call patent dance, because many of these agents have multiple patents on different components or different steps in the process. So in an effort to side rail or delay approval and marketing of biosimilars, sometimes these patents have ended up in court. But nonetheless, these are the FDA-approved biosimilars that will eventually make their way into clinical practice and hopefully into pathways and guidelines. Now, before we talk about the integration in the pathways, I have to address some of these concerns that I've heard expressed by clinical colleagues and probably by patients, but I think that patients are depending on clinicians to guide them and advise them on whether these are truly safe and efficacious to utilize. So there is remaining skepticism about the FDA process for this. And my only response to that is, we've really looked at this, the regulatory process that's put in place, the safeguards, and as you saw, it's not just the biosimilars that may be vulnerable to some changes that need to be monitored and caught early, the originator as well. I think you can think of the biosimilar as the most recent version of the original molecule. So the biosimilar trastuzumabs that have been approved were developed and regulatory approvals based on all these, as I said, more than 60 analytic quality measures. That was not the case back when Herceptin and Rituxan were originally developed. The technology, the analytic capability of companies and the FDA to review these and approve them has vastly improved over the last decade and a half. So in many ways, you could potentially argue that there's more quality assurance behind many of the biosimilars than there is over the originator. Nonetheless, there's some skepticism that remains. There also is this perception that it will be mainly cost that drives this, and I can't deny cost is an issue. It's one of the motivations for developing and bringing these into the marketplace. But at your own institution, I think it's important to put safety and efficacy first, and then cost may come in if there's no difference in safety and efficacy. There's this perception that switching from the biosimilar from a originator to a biosimilar may not be safe, and I think we now have abundant real-world data to suggest that there's not really solid evidence for that. There's concern that payer formularies will require biologic failure before allowing switching. We're still waiting for these interchangeable biosimilars to come along. So thus, ASCO, NCCN, other organizations, including here, are trying to educate professionals about what these agents are, the processes involved, where we are in the approval process, and also their integration, again, into clinical practice and into pathways and guidelines. We've integrated the supportive care biosimilars into ASCO growth factor guidelines. This goes back now 3 or 4 years. And these are being updated, but presumably biosimilars, white cell growth factors, will also be integrated into the ASCO update. They've already been integrated. We just updated the NCCN growth factor guidelines, and all the approved biosimilars are mentioned as alternatives, safe and efficacious choices that you have if you're going to use a white cell growth factor, filgrastim, or pegfilgrastim. So the guidelines have integrated them, and I think it makes sense at this point that pathways likewise integrate these agents into their steps. Obviously, one of the issues is that supportive care biologics or biosimilars is one thing. Anti-cancer biosimilars, where it's treating the cancer and it may be a kind of a life and death issue, may be quite different. Nonetheless, the NCCN Board of Directors, I think 2 or 3 weeks ago, said they have reviewed and have accepted the FDA process for regulatory approval of these biosimilars and are encouraging the guideline committees to integrate biosimilar versions of their therapies into guidelines. This just illustrates the updated NCCN breast cancer guidelines, and you'll see there's a footnote. If you look at the details in this guideline, that's added biosimilar versions of trastuzumab. These will be automatically put in as options for the provider to choose from, in addition to the originator. So ASCO issued this position statement. I was privileged to chair the working group that developed this position statement. This was published in JCO a little over a year ago. And it basically addresses all these things we've talked about, the difference between generic and biologic therapies, what biosimilarity is, the regulatory approval process, and issues like interchangeability and extrapolation. So it's all in there, and I just refer you to that document if you want to read more about it. We highlight in there issues around safety and efficacy, and we came out with the statement, biosimilars will play, if they haven't already, an important role in the future care of patients with cancer and will improve access to valuable medicines. There are the issues, again, around interchangeability that are yet to be worked out. The naming and labeling has created some confusion, but there's a justification for being sure which agent you're using. The value of biosimilars is discussed in this document as well as in the value of cancer care task force documents that have been issued. And of course, there's a need at each of the annual meetings and podcasts in between to continue to educate ourselves and our colleagues and our patients about these processes and provide answers to their questions. So I'll just, this is where I think the rubber meets the road, which is finding the right balance of evidence between biosimilar, requiring the approval process being too onerous, too large, but driving up the cost of development and therefore basically negating the opportunity to reduce prices of these agents if they're approved. On the other hand, if the approval process is too lax or too weak, while the development cost will probably be less, the concern is that healthcare providers may not embrace them and use them as well. And if there's not utilization of these as opposed to the more expensive originators, it kind of defeats the purpose as well. So finding that right balance is critical. I mentioned Europe is out about a decade ahead of us, and I think we should all be reassured that over that decade, they now have 25 oncology biosimilars approved. They have not had to withdraw any of their biosimilars because of efficacy and safety concerns. The EU monitoring for safety has not identified any relevant differences in the severity, frequency, adverse events, and their efficacy. I will mention one caution, which is they now have 10 years of data to show how that uptake distributes across practices. It's quite clear, even in Europe, oncologists and hematologists were more likely to use biosimilar versions of these molecules in later stage disease or in patients not being necessarily treated for curative intent. And it's only over time where the comfort level has been developed, where they're using them up front or using them in curative setting. Nonetheless, over this period of time, no red flags have emerged. So will biosimilars be accepted much like they have in Europe when they get into practice in the U.S.? And how rapid will that be? We really don't know. We have early signs from the growth factors, but it's just far too early. There was a Wall Street Journal op-ed a few weeks ago that said it's time to throw in the towel on biosimilars. in the U.S. And the reality is it's just far too early because we do have a little data on the supportive care biosimilars, but virtually no data on the uptake and impact of the anti-cancer biosimilars on pricing and healthcare costs. And in the end, will these improve access to these therapies, reduce disparities, reduce financial burden? Again, we don't know. The potential is there. This just illustrates that potential and how it's realized will depend on how much price reduction results and how much companies are willing to reduce the price of their biosimilars when they get in the market compared to the originator. So this illustrates the scenarios of 20% price reduction, 30%, or 40%. You can see how much money, $50 to $100 billion, looking at 8 key products in the EU and the U.S., could be saved depending on the degree of price reduction. As I mentioned, we do have some data on the myeloid growth factors, and the top curve just shows the use of filgrastim in the U.S. It hasn't changed very much. Maybe it's drifted down a tiny bit. And the bottom, it shows how the use of the originator has fallen off over the first couple of years through the end of 2016, and the use of biosimilar filgrastim has increased and now is approaching 50% of the market share. And I saw some more recent data for 2018 suggesting perhaps as high as 2/3 to 3/4 of filgrastim use is biosimilar. Again, we'll see where that plateaus or ends up. In terms of cost, again, very early data, only on the growth factors, and it's a complex slide, but just to compare the originator here and either the cost per day or the cost per patient, you can see if you compare that to the true biosimilar filgrastim, there's about a 10% price reduction, ranging from 8 to 12% that's been reflected. Whether that'll be true of all the biosimilars or whether we'll achieve that 20-30% price reduction is yet to be determined. So there are opportunities and challenges here as we begin to close. We want to reduce this unsustainable increase in healthcare costs, and the biosimilars, as I said at the beginning, is one piece of that puzzle. It's not the whole solution, but it may help us towards that goal. And the approval, however, of these agents is based on more limited clinical data, so we have to educate ourselves and our colleagues. We need to monitor these over time. Pharmacovigilance will be key to this. We need to be aware of this issue of drift, variability, immunogenicity, and continue to monitor both the originator and the biosimilar. Interchangeability is yet to come, but probably will come at some point. I will just mention there's an administrative burden. We've discussed that actually over the last couple of days with some colleagues, that at least in our arena, where you have 5 biosimilar trastuzumabs and an originator and multiple insurers, and the insurers often drive the decision on which of these to use, our pharmacy needs to stock all these agents, and that creates challenges, administrative challenges that at the institutional practice level that hopefully will be improved on over time. But I think, again, this comes with challenges as well as opportunities. And I do think it's very clear that high-quality, clinically driven pathways can provide an opportunity to improve the integration of these into clinical practice, improve efficiency and effectiveness of care, whether it's supportive care or cancer treatment, and access to high-quality care at a more reasonable cost. So our opportunities here: additional agents at lower cost, savings and efficiencies to the health system, as well as hopefully eventually to patients, increase access to these exciting biologic therapies, foster innovation. As I mentioned, these analytic care tests that were developed were largely developed as a result of the need to do regulatory approval of biosimilars. So there has been innovation that's resulted out of this whole process, and hopefully will continue to refine the development and monitoring of these agents. And ultimately, we hope we'll see improvement in healthcare outcomes. So biosimilar agents are here to stay. They are out there. Their use is very early. I would say that the rumors about their demise are greatly exaggerated, and they're probably going to be with us, and we need at least, I would argue, 5 to 10 years to know whether we're seeing meaningful reductions in healthcare costs, and whether, again, in the U.S. setting, we, as opposed to Europe, see any red flags in terms of safety and efficacy deviation. The uptake of these will depend on very close monitoring and integration into clinically driven pathways and guidelines. And we need to continue to educate ourselves, and we're charged with doing that throughout the U.S. So that people understand the goals here, the processes, and the value of integrating biosimilars into modern oncology care. Thank you. So I know we're short on time, but glad to take any questions after the session.
That'd be great. I'd appreciate that. Thank you very much for your time.
Sure thing.
Thank you.